is Week 6, Video Lecture 11, Microscopic Life Forms, Part 2. Okay, so we just finished looking at Part 1 of Microscopic Life Forms in class. And so primarily there we were looking at beneficial flora, although we were also looking at opportunistic flora. And certainly there is overlap there between those. This lecture, what we're focusing more on is more on the opportunistic flora and the harmful bacteria, or the harmful flora side of things. So let's take a look at our objectives. Identify symptoms of overgrowth of opportunistic flora. Identify various opportunistic and harmful strains of microscopic organisms that populate the human body and diseases associated with each. So here's this slide that we looked at in class earlier. So we can see that overlap between beneficial flora or bacteria, as they call it here, beneficial flora and harmful flora, and in between there where they overlap, we have this opportunistic flora. And so we know that the beneficial flora is not only fine to have, it's good and important to have around. It helps to maintain proper health and functioning of tissues. And the opportunistic flora is fine to have around as long as it's in the right place and not too numerous, then it's not a problem. Harmful bacteria, however, here's the irony of harmful bacteria. There's that overlap between harmful bacteria and opportunistic flora. And so there are a number of strains of what are considered harmful bacteria that are naturally occurring in our flora. So we'll take a look at that, and we'll but also, like for instance on this slide, they give Clostridium as one of the strains of harmful bacteria. So there are many strains, for instance, of or many species of Clostridium, and some are harmful and some are not. Some are deadly, some are very, very harmful, and some are not. So that's partly why we can see some of these naturally occurring in our biome, and they're only mildly problematic, or they function essentially as opportunistic flora. But some are really deadly if they get on board. All right, so let's just sort of recap where we've been in our little microbiology journey so far. So in the last couple of lectures, we have kind of seen this history of microbiology. We looked at Louis Pasteur and the discovery of microscopic organisms in general, and he found that there were ones that could ferment things, and when they fermented those things, it made those things not only fine for us to drink, but even beneficial for us to drink. And then he also found that there were strains that if they grew in the same substance, they would spoil things, and we'd get sick if we drank that substance after some other strain grew in it. So that was sort of the beginning of yes, there are these microorganisms, yes, they're everywhere, and they serve different functions. And sometimes we don't want them in our body, and sometimes we do. He made links between specific types of microorganisms and diseases, and from that developed vaccines. But also in the process of developing vaccines, he saw that he could manipulate the environment bacteria was growing in, and in other words, change the medium that bacteria was growing in, and come up with a slightly different version of that bacteria, perhaps even more deadly or more dangerous, or really mild and not virulent or dangerous at all. So all of these things were being discovered in the mid to late 1800s. We took a look at viruses and exosomes, and I should say viruses, or AKA exosomes, depending on whether we're viewing those things as viruses or as exosomes. As viruses, we definitely could associate specific diseases with them, and as exosomes, we could associate adaptation and cell-to-cell communication, adaptation to the environment and cell-to-cell communication. So the whole virus end of things we looked at as far as what is a virus or what is an exosome. Is it alive or not? Is it a method of communication between cells? is it something for us to be concerned about? So that's all something that we looked at. And then that moved us on to looking at flora. So if we consider viruses or exosomes to be alive, then we could say that those are part of our flora. If we don't consider them to be alive, then they're part of the molecules of our body. And flora then would be, some of the living things that we have, or all of the living things that are in us, and those would be bacteria and fungi and, parasites, things like that. Okay, so in our last video, our last lecture, we looked primarily at beneficial flora. Now we're moving on to look at the opportunistic or potentially harmful flora. And one of the things that I hope you're getting here as a takeaway is the balance that is required for good health. That we need to have a balance between beneficial flora and opportunistic flora. In many cases, and we'll see some more of this in this lecture, that sometimes the only thing that keeps potentially harmful bacteria in check is the beneficial flora, because a lot of that potentially harmful flora is resistant to bacteria, but it's not resistant to the forces of beneficial flora. So beneficial flora can make an environment for itself to thrive in, and when it's doing that, it keeps the harmful or opportunistic flora from growing. So balance is key. We don't want to have too little of the beneficial flora. We don't want to have too much of the opportunistic flora. We don't want harmful flora that doesn't belong in the body at all. And we want the tissues of our body to support the correct balance that is going to maintain health for us. So let's take a look here at symptoms of overgrowth of opportunistic flora. So again, opportunistic flora is flora that is naturally occurring in our biome, and also in the last class we also looked at dysbiosis, which is an imbalance between flora. And so what we're looking at here, when we're looking at symptoms of overgrowth of opportunistic flora, we're looking at symptoms of dysbiosis. That means symptoms of there being not enough beneficial flora and there being too much opportunistic flora. And what's interesting about a lot of these symptoms is many of these things we don't associate with our flora at all. We just think, whatever. It's just being in a body, it's just life, it's just, maybe I should eat better or whatever. But these are all signs and symptoms that can arise if there is an overgrowth of opportunistic flora. So I'm not going to go through every single one here on this list, but it's a good list to take a look at. So certainly there are some things on this list like itchy anus or vagina and yeast infections. Those things are a little more obvious, like what's going on there. There's not enough beneficial flora in those tissues to keep the opportunistic flora from growing. And so candida, for instance, is an opportunistic flora that will take hold and start to grow in those tissues and cause itchiness and irritation if there's not enough beneficial flora. But one thing that's interesting, for instance, craving foods that feed opportunistic flora, that kind of makes me think, anyway, who's running this show? So we did look at how there are 10 times more flora cells in our body than our own cells. And so it certainly makes sense with those sorts of numbers, if we have a lot of opportunistic flora, that it would have the capacity to put out hey, feed us some bread, hey, feed us some beer, or whatever, something that's going to support its growth. And if you stop giving it, then it's like, hey, where'd that go? We're starting to die off. We want more of that stuff so that we can live well. And so it's sort of it allows us to potentially reframe food cravings. Canker sores, when we rethink of these things, we go, oh yeah, I guess that could totally be considered an overgrowth of opportunistic flora. It's a little infection, an infection in the mouth, and Most likely it's an infection of something that has been there all along. It's just the opportunistic flora has grown out of control. The beneficial flora has taken a hit for some reason, and we looked at in the last class at some things that could make that beneficial flora take a hit, like stress, for instance, is one. And that's a great example with canker sores. If you have ever gotten canker sores, if you recognize that very often they come up more often and more frequently during times of higher stress. So that higher stress is decreasing the health of the beneficial flora, and then that gives an opportunity for opportunistic flora to take hold and start growing more. Stomach ulcers are associated with H. pylori. H. pylori is in that group of opportunistic, potentially harmful actually. But it's we have it around quite often. It doesn't necessarily have to be with us, but it is associated with things like stomach ulcers, and which is kind of funny because where stomach ulcers happen in the stomach, and we kind of think of the stomach as an environment where a lot of things, how could something survive the environment of the stomach? Well, there are certain bacteria that is exactly the environment that they want to survive in. And so H. pylori is something that can grow there and can grow and can develop stomach ulcers if they do. All right, let's see. So let's go on. So I just touched on a few of these, but you can look through those and kind of see if they make sense to you. And some more of these will probably come up as we go through this lecture today. So one of the things that Louis Pasteur kind of left us with, is that killing the opportunistic flora alone often does not eliminate the condition. Now, I don't know that he said that, but he would say, oh yeah, that completely rings true. But we know this, right? If you've ever, say, had a yeast infection or had athlete's foot or something like that that's considered some overgrowth of something, and you put something on it to kill it, well, that works, right? You put that cream on and it kills it and it's gone. And then you stop using the cream, and what happens? Very often it comes back. So just killing that opportunistic flora, just killing it often does not eliminate the condition. Why? Because we haven't changed the medium. And if it's still a medium where that flora can grow, it's still going to grow. So changing the medium resolves the condition. This is sort of the take home that Louis Pasteur was trying to get the medical community to understand, that they were not so interested in understanding, but changing the medium resolves the condition. So what does that mean, changing the medium? That means changing the stress levels, changing dietary habits, changing sleep patterns, changing whatever it is that is causing some sort of compromise in the tissues that is compromising the health of the beneficial flora and supporting the health of the opportunistic flora. So we hadn't named sort of an alternative theory to the germ theory, and I think we've seen evidence to say, okay, germ theory is valid. There are these things that you're not going to develop strep throat without the streptococcus bacteria. It's just not going to happen. However, the flip side of that is not everyone who comes into contact with the streptococcus bacteria will develop strep throat. So anyway, germ theory tells us that that microorganisms cause disease. Terrain theory, so that's sort of an alternate theory to germ theory, tells us that it is the medium that is important in whether or not a disease is going to thrive, or that whether or not a microorganism can live in that tissue. So germ theory would say vaccinate the fish so it can survive in a dirty fish bowl. Terrain theory would say clean the tank, make the medium something that anything that's going to harm the fish won't be able to survive in, and then the fish will live a lovely life in a nice clean tank. So anyway, so that's an alternative, and probably in reality something that is kind of between the two of those, or a balance of both of those ideas is probably something that's going to answer a lot of questions about causes of conditions and what can we do about how can we treat conditions. Okay, so let's start going through some of these microorganisms that are more strongly associated with disease or imbalance in the body, and can give rise to various not so pleasant things. So the first one we're going to talk about here is Candida. So Candida albicans is a fungus, and it is a normal part of our biome. So on the notes on the bottom of page 2, you'll see the Candida part of the chart. So on the left of the chart is the name of the microorganism and what type it is, etc. So this is a fungus. There are over 20 different species of Candida. So Candida albicans, though, is one that that hangs out in the body and can become problematic. Some strains of Candida are resistant to antibiotics. So this is the very problem that we have when we try to only address these things with germ theory, because germ theory says, well, if we kill it, then we won't have the disease. But we know that if all we try to do is kill it, it doesn't always work, right? If we are just using antibiotics, what we develop is so what we're doing is we're changing the medium a little bit, and the surviving bacteria or whatever, in this case a fungus, they're ones, using antibiotics helps us actually to develop and grow antibiotic resistant strains of things, which is really less than ideal. Anyway, so that's on the left side of the chart. On the far right side of the chart, it tells us where this fungus comes from. How does it get into our body? Well, Candida is ubiquitous, meaning it's around us everywhere in our world, but it's also in us and in and around our bodies. So it is part of our natural biome, and it's part of our skin. It's on our skin. It's part of that flora, and it's in our gut flora. So it's most closely associated with skin, with mucous membranes, and with the GI tract. And there are some conditions. This is what it looks like. This is Candida under a microscope. And there are some specific conditions that are associated with. And these are not life-threatening conditions, but they're not comfortable to live with either. So one is thrush. Thrush is more common in nursing infants, or actually they don't have to be nursing infants. It's more common in infants, and it's more common in older folks. This looks to be like a kid that's not an infant, but this kid has thrush. So this is a thick growth of Candida on the tongue. So that's thrush. Another way that Candida shows up is as skin infections. And that will tend to be in warm, moist areas like under the breasts, in the armpits, in the groin region, or like in folds of skin. Let's see what else. But then the worst thing though about Candida, the most dangerous expression of it is invasive candidiasis, which is when Candida gets into the bloodstream, and then it's very difficult to treat because it's resistant to antibiotics. And really the primary way to treat it, so Candida is something that feeds on sugars, and so if people have a Candida issue, Candida overgrowth, anytime they have anything with fairly simple carbohydrates in it, the symptoms get worse. And so when Candida gets into the bloodstream, we always have some level of glucose in the bloodstream. So it's like, woohoo, here we go. We have everything we need to live a nice, happy life here. And so it can be very difficult to get rid of it. It's resistant to antibiotics, and basically the thing that we can do to get rid of it is keep our blood sugar as low as possible, and not eat things that make it grow. But it takes a long time to get rid of it. So it can be really tough to treat. So that's Candida. That's a fungus that we have in us all the time. And I didn't say yeast infections, did I? That's a super common expression actually of Candida. Candida is what is the flora that overgrows very often with vaginal yeast infections. Okay, let's look at Clostridium. Clostridium is a whole genus within which there are about 100 different species. And these are bacteria. And some Clostridium species are resistant to antibiotics. But there are, so a whole lot of, the vast majority of the Clostridium bacteria don't really want to have anything to do with us. They can't live in our bodies, and they don't really care about us at all. But there are 5 strains or 5 species of Clostridium that grow quite well in the body, unfortunately, because they're not so fabulous. But here's what Clostridium looks like. And so the 5 that, so on the chart, if you look at the chart, what we see is, so like the first one is C. botulinum, which is so that's Clostridium botulinum. It causes botulism. And so we've probably heard of botulism before. Botulism is not a disease that we have around so much anymore, but it's still, I mean, it absolutely can happen. It's just that we know how to prevent it. It can show up in canned goods and in honey that hasn't been packaged properly. Canned goods that have, that haven't been properly boiled and sealed and stuff. And so if oxygen gets in there, botulism somebody could get botulism by, so C. botulinum can grow in there. But what do we use it for? Very commonly today we use the toxin from C. botulinum, and that is what is in Botox. That's what Botox means and stands for, is the botulism toxin. And so botulism as a disease is dangerous because it causes paralysis. And so that's exactly what we use the Botox toxin for, or what we use Botox for, is paralysis. So we inject a small amount of it into the muscles of the face, and you can see on the before picture that there are wrinkles around the eyes of this woman, and in the after picture there are not wrinkles, because why? Did we iron out the skin? No, we paralyzed the muscles underneath, and that's how Botox works. Very, very popular treatment these days. And it only lasts for a while. It will eventually, go away. So in, a number of months, she'll need to get another Botox injection if she wants those wrinkles to stay gone. C. difficile is one that we do have in our gut all the time. It's a normal part of our gut flora. It is one that is resistant to antibiotics. So, and this is the one that is associated with antibiotic-related diarrhea. So, very often when you take antibiotics, essentially what's happening is you're killing most of the flora in the gut. You're killing everything, all of the beneficial flora, except for what's in the appendix, and you're killing a whole lot of the opportunistic flora also. But you're not killing the C. difficile because that is resistant to antibiotics, and so that thrives during that time when the rest of the flora have been killed off in the gut, and that causes the diarrhea that's associated with taking antibiotics. Perfringens causes food poisoning and gas gangrene. I did not include a picture of gas gangrene in the slides for this lecture. You're welcome, but you can look it up if you want. It's super gross. And can be fatal. Perfringens, I think I'm saying it close to correctly, is in the soil. And so it enters through significant skin openings. This is the kind of thing like on battlegrounds in the old days, when the soil was bursting up with explosions and people's bodies were exploding and dirt was getting into wounds. This is the kind of thing that would. And it's still in soil, and this can still happen, which is why you can find pictures on the internet of it, but, only for the strong-stomached viewers. C. tetany, we've heard of that guy before, right? Because it causes tetanus. And, tetanus is something that it's part of normal vaccines, getting vaccinated against tetanus, getting a tetanus shot. It's a rather uncomfortable vaccine, as vaccines go, but tetanus causes paralysis and rigidity and causes death, which is why there's you hardly ever, in your practice likely you will not see tetanus. It's not really that relevant in today's world, but in theory it's still around, and basically it also, like the one that causes the gas gangrene, it happens with wound contamination. So it can be in dust, in soil, in rust, like, you know, that kind of thing. So like if you step on a rusty nail or something, that's kind of the thing that, like, oh, we need to make sure we get a tetanus shot if that happens, because we don't want tetanus to occur. All right, so let's see what else. And then the last one is C-sore deli, which only really shows up in hospital settings. And again, things that show up in hospital settings, that means it's resistant to antibiotics, because that's really the only thing that can live very well in a hospital setting or a very, very sterile environment, is things that are resistant to that sterile environment. So it really only shows up. People get this infection following a medical abortion. So very rare, not a common one that we'll see very much. But those are the 5 strains of Clostridium that are specifically associated with diseases in humans. Let's look at Staphylococcus. So Staphylococcus aureus is a specific bacteria, and this is commonly found on our skin and in the respiratory tract. Here's what it looks like. Kind of looks like bunches of grapes. This is the one, if you hear about somebody having a staph infection, that's Staphylococcus aureus. And it's tricky to deal with because, why? Well, because it's resistant to antibiotics. So we have it around. It can show up. I think I didn't also include a picture of this one. Did I? No, I didn't. I didn't include a gross picture. Again, you can find gross pictures if you want to. It's associated with MRSA. Or it is MRSA. MRSA is methicillin-resistant Staphylococcus aureus. So the SA of MRSA, that's Staphylococcus aureus. That's this particular bacteria. Methicillin-resistant means that it is resistant to antibiotics. So, and so this is the guy that causes, again, a lot of problems in hospital settings because. And so what can happen is, like, there can be infections of, like, wound sites, or infections of like an IV site or something like that. And so when an infection happens in a hospital setting, it's likely an infection by something that's resistant to antibiotics, and Staphylococcus is one of those guys. So let's see. Anything else about that? Okay, I think that's good with Staphylococcus. So MRSA and Staphylococcus, they go together. They are the same thing. Streptococci. So Streptococci is another genus. So it's a whole group of bacteria. There are 50 different strains of Streptococcus, and they are not all pathogenic, and some are pathogenic. So what's curious, if we look on the right side of the chart, so non-pathogenic strains are ubiquitous in the human body. So we have plenty of Streptococci strains within us. There is a specific Streptococcus strain that is a necessary ingredient in Swiss cheese. So these were, you know, some of the things that that Louis Pasteur was figuring out, like, oh, this is the bacteria that turns milk into this kind of cheese versus that kind of cheese, or turns grape juice into this kind of wine or that kind of wine, or whatever. So there is a Streptococcus strain that is a necessary ingredient for Swiss cheese. Pathogenic strains of Streptococcus tend to be more problematic in children and in the elderly and in alcoholics and in people with compromised immune systems. So what sort of issues do we get from Streptococcus? Well, it probably sounds familiar, and it's because it's associated with strep throat. And other conditions. So strep throat is a very common one. Pink eye also is a Streptococcus infection in the eye. Bacterial meningitis is usually Streptococcus that causes that, as is bacterial pneumonia. So these are all, like, they're not so terribly uncommon diseases. You know, will you see these in your practice? Strep throat, likely you'll see. Pink eye, good chance you'll see. Meningitis and bacterial pneumonia, probably not, but you never know. Anyway, but if we see somebody in our practice and we suspect that they have strep throat, and how would we know? Because of the white exodus on their red swollen tonsils. That's kind of the hallmark sign of strep throat. This is something that we want to refer people out to Why? For antibiotics. Even though we know those antibiotics are going to kill everything in their gut, we're also going to have them be taking probiotics while they're taking those antibiotics. But why do we want to suggest antibiotics for somebody with strep throat? Because that very bacteria, if it gets deeper into the body, can cause meningitis, can cause bacterial pneumonia, can cause endocarditis. So those are conditions that are far more severe than strep throat. So it's kind of good to nip it in the bud and make sure it doesn't get past the throat and get into other tissues of the body. And even there's a strain of streptococcus that causes necrotizing fasciitis, which is a flesh-eating bacterial infection. Good times. Okay. E. coli is our next one. E. coli, I know that you've heard of. It's a very, very common guy. And again, not all strains are pathogenic. E. coli is actually a great example of a opportunistic bacteria, because it is in us all the time, but it just needs to be in the right place. And when it's in the right place, then it's beneficial, actually. It's doing good things. When it's not in the right place, that's when it causes problems. So the pathogenic strains of E. coli, where do we get them? We get them through fecal-oral routes. So because it is supposed to be in the colon, and that's fine for it to be there, but it's when we get it into other parts of the GI tract, like in the stomach and in the small intestine, that it could be problematic. Or if we get a strain that doesn't do so well in our body. But E. coli strains make up part of the healthy flora of the large intestine, and these are the guys that make vitamin K. And let's see, but pathogenic strains can cause severe food poisoning. So this is what E. coli looks like. And of course, this isn't really what it would look like if you looked under a microscope. They make these look really clear and easy to see. They're, these things are not really very easy to see under a microscope, but it's kind of nice to see a really clear picture of them. And so our last one here is Salmonella, which again, I know that you've heard of. There are over 1,000 different host-specific strains of Salmonella. So meaning there's Salmonella that lives in chickens, and there's Salmonella that lives in cows, and there's Salmonella that lives in rats, and there's Salmonella that lives in humans. And so when we have a problem with Salmonella is when we get some strain other than a strain that we want to have in our human bodies. That's what causes the illness from it. And Salmonella, where have you heard of it? You hear it on the news from time to time when there's a food poisoning in some area, like, the onions in this restaurant turned out to have Salmonella. The romaine lettuce turned out to have Salmonella. So usually what happens with something like that is maybe it was in a fertilizer that was used on that food when that food was growing. Most likely if there's a large contamination of it, like a whole field of lettuce or something like that. And so then, but with Salmonella, typically it's not a big deal for healthy adults, but it is a big deal for younger kids, and it is a big deal for older folks or for people with compromised immune systems. Also, it can be in food, and it's not a big deal if we cook the food, because it's not going to survive that cooking. And so the problem with Salmonella comes with raw or undercooked food. But it causes food poisoning, so that's going to look like diarrhea, vomiting, fever, abdominal cramping. Like, not a fun ride, not something we really, want to do if we don't have to. Okay, so that is our brief look of a very few common bacteria and one fungus that, when they show up in the body, can cause particular problems. So this was our look at some harmful flora and some opportunistic flora. So we looked at symptoms of overgrowth of opportunistic flora, and we looked at various opportunistic and harmful strains of microscopic organisms that populate the human body and diseases associated with them. So your homework, oh wait, do you have homework? Well, I guess, let me think here. What is your homework? Oh, I know what your homework is. It's posted on Populi. It is, you are going to go through the strains that are listed here. If I remember correctly, you're going through the strains that are listed on the handout. I'm pulling it up on Populi right now on my computer, and I'm reminding myself what it is. you are, yeah, you're going through the strains, and you're indicating whether it's a normal part of our biome or not, and if not, you're indicating where or how a person might acquire it, okay? So that's all right there on the chart. You're basically going to go through that chart and say, oh, okay, Candida, this is a normal part of our biome. Or, you know, Clostridium, there are strains that are a normal part of our biome, like C. difficilis, but there are strains that are, for instance, we would get it, you know, it's in the soil, we'd get it through wound contamination. So that's essentially what you're going to go through. Where do these strains come from? Where are they hanging out if they're not in our body? And how do they get in there? Or if they are in our body, tell me that too. Okay, so that's it for homework for this section. And that's it for this whole microbiology section as well. So next up is you're going to go on to Populate, complete exam 3, and then you're going to go on to video lecture 12, which starts a new topic, which is not completely new because we've already had an introduction to cells, but now we're getting into animal cells. So we're going to start to go through the parts of a human cell, and the next video is just about, you know, the functions of the different organelles, and then after that we'll take a look at the cell membrane, the structure of it and the function of it, and then we'll start to get into different specific functions that cells carry out, like I think our first thing is ATP production, which is a really big deal of one of the big important things that cells do. Okay, so that's it for this lecture. I hope it was enlightening and entertaining and everything that you hoped it would be. I will see you in class very soon. Have a fabulous time until then.